transitions out of it. And from looking at several cross-sections throughout the length of the same lateral line nerve, we could actually tell that this happens in an anterior-posterior fashion, such that portions of the nerve that are closer to the cell bodies of those axons are already located in their mature position below that epidermal basement membrane. Those that are closer to the outgrowing axons are still located within the epidermis. And those in the middle are in this transition state with basement membrane both above and below the nerve. And so this actually became very interesting in light of results that we had in the lab about the localization of the lateral line nerve in mutants that lack Schwann cells. So here again is just the wild type nerve, and the lateral line nerve is below this epidermal basement membrane. But when I was looking at 3 different mutants that lack Schwann cells, ERB2 and ERB3 mutants where Schwann cells are present at the ganglion but fail to migrate out, and these guys will come back later, and colorless mutants, which are mutant for SOX10, so they have no Schwann cells whatsoever. In all of these cases, I always found that the lateral line nerve was aberrantly localized within the epidermis above this epidermal basement membrane. Additionally, it was also often located near neuromast cells labeled by ends. And so I think when we saw this defect initially, we just assumed that it was an axon outgrowth defect. So to figure out whether it was that or perhaps that Schwann cells were actually playing a direct role in this process, we looked again early in development while the axons were growing out in both heterozygous siblings and mutants and found that actually axon outgrowth is indeed normal. So at early stages, both the heterozygous sibling nerve and the mutant nerve are both present. They're located within the epidermis, as we would expect, above this epidermal basement membrane. And actually, in both cases that I'm showing you here, they're localized in a cluster of pre-neuromast cells. So to get at whether Schwann cells are really what's required to move the lateral line nerve across that epidermal basement membrane, we wanted to create genetic chimeras. And I could not have done this experiment without the awesomely fantastic Julie Perlin. And so what we did was we took wild-type cells that were carrying a FOXD3 GFP transgene. So any Schwann cells that arose from those wild-type cells would be fluorescently labeled with green. And we transplanted them into SOX10 mutant hosts, so no Schwann cells whatsoever normally. And then I screened for the presence of genetic chimeras where there were wild-type Schwann cells in an otherwise mutant background. And then I can look and see where the lateral line nerve is located when we put wild-type Schwann cells back in. And so just an example of what I was looking at here. So this is a wild-type embryo carrying a FOXD3 GFP transgene. And so you can see fluorescently labeled Schwann cells running the length of the lateral line nerve. And then this is an example of a full-length clone of wild-type Schwann cells that have been transplanted into a SOX10 mutant host. And then when I looked by electron microscopy to see where those lateral lines were located, I was very excited to see that when we put wild-type Schwann cells back in, we can fully rescue the position of the nerve so that it's now below this epidermal basement membrane in its proper location, and we can also rescue the myelination state of the nerve. We also had cases where Schwann cells only covered part of the nerve, so this was a smaller clone of wild-type cells that had been transplanted in. So there are axons present along the whole length of the fish, but Schwann cells only make it about halfway. And this allowed us to ask whether the presence of Schwann cells here would rescue the entire nerve or whether they could only rescue portions that they were directly associated with. So when I look at this anterior portion where Schwann cells are present, we find that the lateral line nerve is, as we would expect, properly located below this epidermal basement membrane. But in contrast, when I look at the posterior segment where there are no Schwann cells, we find that the nerve is still aberrantly localized above this basement membrane within the epidermis. So Schwann cells can only move portions of the nerve that they're directly associated with, suggesting that this process is maybe regulated by short-range cues. So this was really exciting. We have Schwann cell-less mutants, the nerve doesn't move. Put Schwann cells back in, it does move, but it wasn't actually that perfect all the time, and that turned out to be really informative. So this again is just this case of full rescue, where the nerve is totally below the basement membrane. But we also had cases where the nerve got caught between 2 basement membranes, suggesting that the Schwann cells that were transplanted in could make a new basement membrane, but they could not degrade the old one. We also had cases of partial basement membrane degradation, so the nerve is properly localized below this epidermal basement membrane, but there's still old basement membrane remaining, again suggesting that the Schwann cells were not able to completely degrade that old basement membrane. And finally, my favorite case was we had an example where the entire nerve, the Schwann cells and the axons, were localized within the epidermis, but those Schwann cells were actually able to induce the formation of an ectopic basement membrane that went circumferentially around the nerve. So this suggested to us that maybe Schwann cells can always make a new basement membrane, but they can't always degrade the old one. And so at this point, we were really wondering, why does the lateral line nerve need to move at all? It grows out within the epidermis, its targets are within the epidermis. Why move it across that basement membrane at all? And the answer came from just looking at a time course of wild type and mutant nerves. So these are lateral line nerves that have been labeled by an antibody against the cellular tubulin. So the axons are labeled fluorescently, running straight down the length of the fish. And then at this early stage, at 2 days, we find that the neuromasses of the lateral line, which are labeled by arrowheads, are usually located very close to that lateral line nerve. But by 3 days, in wild type, these neuromasses will begin to migrate ventrally away from the lateral line nerve, so that by 6 days, there's actually a significant distance between the main bulk of the lateral line nerve and the location of these neuromasses. Individual axons will peel off to innervate those neuromasses, but the main bulk of the lateral line nerve really remains anchored in place. And in comparison, when we look at mutants that lack Schwann cells, so I'm showing you here ERB2, but the same is also true of ERB3 and SOX10 mutants. So again, at this early stage, the lateral line looks normal, so the nerves are running straight down the length of the fish. But beginning at 3 days, again, when in wild type, these neuromasses begin to migrate away, we begin to see significant disorganization of the nerve. The nerve begins to buckle. We see individual axons fraying away from the main bundle of the nerve. And also neuromasses, which are normally always located ventrally to the nerve, can now be found dorsally to the nerve. And so what we think is happening is that because this nerve remains aberrantly localized within the epidermis, that as these neuromasses begin their ventral migration, they're actually pulling the lateral line nerve with it. And that this pulling force is what's causing the disorganization that we see. And it's also causing kind of shearing pressure on the nerve, which causes it to fray and individual axons to be peeled away. So we think that actually moving the lateral line nerve out of the epidermis serves a very significant protective function. And so just to give you a sense of what this looks like again by electron microscopy, so here's a neuromass, and a mutant nerve is located right within the center of it, and then you can see that there are individual axons that have been pulled away from this main bundle. But by contrast, in wild type cases, the nerve is located all the way down here, and the neuromass is all the way up here, so there's a significant distance between the two at this stage, and they're separated by this epidermal basement membrane. And in this case, we've actually caught a branch of the lateral line nerve, recrossing the basement membrane to go and innervate that neuromass. And I just wanted to remind you again that we know that extensive growth of the organism and neuromass addition and rearrangement occurs throughout the life of the fish. So we really do think that this is a way that the fish can initially lay down the nerve, but then take steps to move it away from the reorganization of its target and really sequester it from the continual development of the organism. So just to quickly sum up what I just told you, I told you that we found a new role for Schwann cells in localizing a peripheral nerve. We found that the lateral line nerve initially grows out within the epidermis, but then is rapidly transitioned across an epidermal basement membrane. And that Schwann cells are required for this process. So when Schwann cells are absent, the nerve fails to move. And that this results in significant disorganization of the nerve.